So, yeah, I think it should be fine now. Okay, so hello and welcome to the new course on genes, genomes, and the epigenome. This will be quite a long course, which will stretch over a span of around 50 hours, 48 to 50 hours, which will split over a large number of weeks, around 20 to 25 weeks, where we'll be discussing a large number of topics, some of which you already are familiar with, which we'll brush up on, and we'll introduce some new topics, some current high throughput technologies that are increasingly becoming very prevalent in the research sector in academia so that you become abreast with it, and will also understand some basic tools that are used to analyze the same. So just for people who have, because a lot of people have not joined, and for people who have already been a part of my previous courses, you would know that these lectures get uploaded on YouTube. So you can access it from there. And also, once I get the link for the course portal, we will also, you'll also get access to it there. Additionally, we will be having some assignments or some exams during the tenure. Uh, Nothing too serious, just some quizzes so that you can refresh your memory regarding the topics that have already been covered. Now, before we proceed further, just as a brief introduction, my name is Ankita. I am a PhD student at IIT Kanpur, and my research work centers majorly around the topics that I'll be teaching you. I work in cancer biology, so we are actively involved in learning about the genome, how it functions, how it is regulated, what are the different ways in which changes in the genome can affect a large number of diseases in the long run. All of that information is something that we are very actively involved in studying about. Hence, my course is centered around these topics. Now, first, let us understand the overall outline of the course and how we'll be dealing with it. So, this is the flyer, which you would already be familiar with. Let me just get the laser pointer. Yeah. So this is the flyer, and as you can see, we will go over a very large range of topics, starting off with gene structure, function, and regulation, which is the topic we'll be covering today. And we'll cover a part of it today. Majorly, we'll be brushing up on some initial ideas that you must already be familiar with if you have had some basic biology experience. If not, this will be a quick revision for you, and also how basic gene regulation is carried out within cells, irrespective of whether it is a normal cell or it is the physiological regulation of a cell under different circumstances, whether it is stressed or it is deprived of nutrients, etc. We will be going into a little more detail about gene regulation in the next week, where we'll be learning about a detail about methylation and how remodelers, etc., also have a role to play in the same, because that also overlaps with another topic that we have in the upcoming weeks. But majorly, this will be the idea we'll cover for today. Moving forward, we'll learn about mutations, the different types, how they're characterized, how they were first identified, what effect does it have in the long run, why at all are we not able to cope up with mutations, and why are they such a huge problem? How are they affecting us in the long run? What is the difference between a mutation and a polymorphism, and how functional genomics, which is quite actively taught as a subject in a large number of courses, how that is actually relevant in the real world, and whether or not we can implement it for our own good. Um, Moving forward, we learn about sequencing methods. You would probably already be familiar with Sanger sequencing, which is the most, I wouldn't say primitive because it is still done till this date, but it has undergone a large number of changes to make it more, um, more relevant in the current scenario. And also, we'll be discussing about NGS sequencing methods, which are next generation sequencing methods, whereby which is what is actively done in today's time, where we learn about the genome or about the transcriptome, methylome, or the proteome for that matter. It's not exactly a sequencing technique, but it is a high throughput method where we study what is occurring in a large number of cells or in a single cell in one snap. So you get a cohesive outlook of what is going on inside a cell under a certain circumstance and use that to draw certain conclusions. So that is the idea behind next generation sequencing moving forward from Sanger. Following that, we will learn a little more about the genome, exome, and the transcriptome, which is going to slightly overlap with the topic that we will be introducing today. But please understand that these are limited. There's very limited time in each lecture to cover everything, so we have to cover some topics and some in other. But overall, you'll get a comprehensive view of everything that you should know about these topics. Following that, we'll learn how high throughput sequencing data is analyzed. Of course, not the coding part, that is not going to be under the purview of this particular course, but we'll learn what are the common tools, what is the biological information that is relevant to be drawn out as a conclusion of this analysis. Because technically, if you give a code to any person who knows some basic, who knows how to handle most of these who knows Linux systems, who knows how to use R, anybody would be able to run those codes. But how to make that biologically relevant, how to understand and make sense of the information that comes out of that analysis is what we'll try to keep our focus on. Following that, we'll understand how cancer genomics has integrated NGS and how it has become a huge arena that has exploded in the past decade or so, how therapeutic targets and disease drivers are being identified, how it is still relevant, even though it is a decade old method, how we are still modifying it and trying to derive more relevant information from it. We'll understand how epigenetics has a role to play in this. We'll look at some key concepts. We'll understand DNA methylation and histone modifications as part of epigenetics. We'll understand how DNA methylation is mapped to decode the genome. What are the high throughput methods that are used to decode the same? We'll see how DNA protein interactions are also relevant, which is where we'll switch to ChIP-seq. So as you can see, this course will cover a large number of different techniques, starting off with Sanger, RNA sequencing, that is whole transcriptome sequencing, single cell RNA sequencing. Following that, ChIP-seq, ATAC-seq, which is for chromatin accessibility profiling, Hi-C, and we'll also look at how clinical diagnostics and therapeutics are enriched by the invention of these techniques, how they have helped us to figure out epigenomic markers and how they are used for testing. And it is actually something that is being actively researched in today's time. It is considered to be translational and therefore is very relevant. And finally, how integration of all of this is extremely cumbersome as a method, but can give you very meaningful results instead of just focusing on one type of method and drawing very strong conclusions about the same. So that's the broad outlook of this course. A lot of things to cover, but we'll take it at a slow pace. We'll try to cover as much information as possible in each class, and hopefully we'll learn something useful as an output of the same. So before we move towards all of these fancy techniques and all the jargon that is going to be very relevant eventually, we need to have our basics very clear because if you don't have this foundation clear, if you don't understand the DNA right now, you will not understand how these sequencing methods work and why knowing basic biology is very relevant for it in the long term. So today will be more of a recap class, but also very foundational in nature. So overall, this is how the topics are kind of spread out. The discovery of DNA, unraveling its structure, how genome is organized, and how is gene function regulated. So these are the topics we'll be covering. I will switch off my video temporarily to make it easier for the recording to be saved. If there's any doubt, if you cannot hear me or anything you want to discuss, please interrupt me at any point of time. Feel free to do so, and we can take the discussion forward accordingly. Okay, so this is the topic for today where we are going to discuss the gene structure, function, and regulation. So starting off here, the topic that you can see we'll be discussing initially is the discovery of DNA. Here you'll see I have I have put in the images of some very renowned people, some stalwarts in this field, Frederick Miescher, Oswald Avery, Colin MacLeod, Mackie McCarthy, and Frederick Griffith. So why I've put this is because I want everybody to be familiar with the people who made these incredible discoveries, and with very limited resources, they were able to draw such foundational conclusions that have changed the force of nature. Nature in the sense that we have been able to understand DNA, modify it, and use it to our own convenience in today's time. So some of the initial experiments that were carried out that are considered as these landmark experiments include the biochemical investigation of DNA. Now let's get some perspective about it. Initially, what was understood about the DNA was that there is some sort of a master molecule that is able to carry information on from one cell to another. But there was no evidence that could support what was the molecule that was able to do so, whether it was retained within the initial cell that passed it on to the next, or whether it was lost, whether or not it was undergoing some modification. None of this was known. So, one of the preliminary experiments that tried to delineate the biochemical nature, when I say biochemical, it means what sort of a molecule is it that is actually able to do so, was carried out by these few scientists that I have mentioned at the top. So in 1868, Frederick Miescher, he carried out an experiment where he could figure out that it is possibly DNA which stores this genetic information, that it is some sort of an acidic molecule made up of nucleotides that is able to function as this hereditary information carrier. So Frederick Miescher isolated a phosphorus-containing substance from which he called as nuclein. So this was the preliminary idea, and this was called nuclein because it was isolated from the nuclei of pus cells or leukocytes, which he obtained from discarded surgical bandages. So he found that nuclein consisted of some acidic portion and a basic portion, so that he considered as protein. Again, this is a hint about what was going to be discovered several years later, that DNA is not just present in the cell as a free molecule. It is present in a very tightly packaged and very well regulated manner, very well structured manner, as a DNA protein complex, which we in today's time understand to be nucleosomes, or eventually to form the chromatin. So, sorry, excuse my throat. What he also saw that a similar acidic substance could also be isolated from the head of sperm cells, which, as we eventually understand in today's time, is the information carrier from the male counterpart that helps in transferring information from the father to the progeny, and depending upon what is the type or the quality of the sperm cell, information may be corrupted or may be retained as per the original cell. So Miescher and several other scientists, his collaborators, suspected that nuclein was possibly associated with this inheritance property. So, so this was the initial idea. Next came the experiment by Oswald T. Avery, Colin MacLeod, and Madeline McCarthy, the three scientists shown here at the center. They actually produced the first evidence that DNA is indeed the bearer of genetic information. Now, look at the time gap between these, almost around 70 to 80 years. It took that long a period of time to bring substantial evidence to convince the scientific community that this is possibly the molecule that is uh, our molecule of interest. Let's put it that way. So what they found was that DNA extracted from a virulent strain of the bacterium Streptococcus pneumoniae genetically could transform the non-virulent strain of this organism into a virulent form. What does this mean? So let us look at the images to understand this a little bit better. So again, keep in mind, this is just a schematic representation. So if you take a vial where you have live virulent bacteria, in this case, the bacteria was Streptococcus pneumoniae, which causes pneumonia. So it is a pathogenic form or a pathogenic variant of the bacteria. When this is injected into the animal model, which is a mouse in this case, it was dying. So this injection showed lethality, whereas if you have a non-encapsulated form, which was considered to be avirulent. Avirulent or non-virulent means it is non-pathogenic. It is also a virus, but this virus does not contain the component that causes the disease or causes the infection. So when this was transformed, then what happened? Transformed doesn't, when this was injected into mice, the mouse was alive, which indicated that there was some difference between these two, which is causing this difference. Now, when this live encapsulated virulent bacteria was heat inactivated, which means that it was it was undergoing some sort of a change where experimentally it was subjected to a lot of heat, then that led to the formation of heat-killed virulent bacteria. When this is injected into the mouse lives. That means there was no lethality in this case. However, when this heat-killed virulent bacteria was mixed or was somehow yeah, let's just call it mixed, was mixed with live non-encapsulated non-virulent bacteria, and you now have a mixture where both these types of bacteria are present, then that means there is some sort of an exchange which is taking place between these two molecules, and when this is injected into mice, these mice die. So Frederick Griffith also contributed to similar findings where he had shown that adding this heat-killed virulent bacteria to live non-virulent bacteria permanently transformed the latter into lethal virulent strains and were able to cause lethality again. So there was some sort of an exchange that was occurring between the two. These were the initial findings that were taken into consideration. Oh, sorry. Next, what happened? Avery et al. They explored this a little further. They extracted the DNA from heat-killed virulent pneumococci, removed the protein as completely as possible, and added this DNA to the non-virulent pneumococci. So this is a schematic representation of the same. You have live non-virulent bacteria, and you have the DNA which is isolated from heat-killed virulent bacteria, right? So you have live, non-encapsulated, non-virulent bacteria. So what they did was when they mixed it with encapsulated ones, they saw that the mouse was able to die. So the mice died basically. So what they saw was that DNA was able to gain entrance into this non-virulent bacteria and was able to transform it into a virulent strain. So in case this got confusing, let me just rephrase it once again for ease of understanding. If you have dead bacteria and live bacteria, you have two different vials. Live bacteria obviously was able to cause disease. Dead bacteria initially obviously was not able to do the same. Now, when you are mixing the DNA that you have taken from the dead bacteria and you are mixing it with non-pathogenic living bacteria, then this DNA was able to enter into this non-pathogenic strain and was able to transform it into the pathogenic variant and was able to cause disease, as a result of which the mice died. So this was the observation that they found. Therefore, Avery and his colleagues concluded that DNA extracted from this virulent strain was able to carry this inheritable genetic message for virulence. However, several people were also skeptical about this conclusion and they thought that the protein contaminant might actually have a role to play, which obviously is a very valid rebuttal to have in this case. But how could they actually counter it? They found that A, treating the DNA preparation with proteases did not destroy its transforming activity. So when proteases are introduced into this admixture, then obviously the protein moiety, which is the encapsulated part of the bacteria, uh, when that is degraded as a result of protease treatment, transformation or this ability of a virulent bacteria to gain virulence, that ability was not affected. Second, treatment of the same with deoxyribonucleases or DNases did actually destroy the transforming activity of DNA preparation. So when DNase treatment was carried out, this transformation ability was affected or was lost, whereas when protease treatment was carried out, no change could be seen. Therefore, this was the first very concrete evidence that the scientists were able to show regarding the nature or the biochemical properties of the molecule that carried heritable information within cells. Coming to the second experiment, which was another, um, it's actually a very huge experiment that confirmed these initial findings and is a very popular experiment known as the Hershey-Chase experiment. So Alfred D. Hershey and Martha Chase, the scientists again shown here at the top, they performed an experiment that provided independent evidence that it is DNA that carries genetic information. So what did they have in this case? Again, understand this very carefully. Please let me know if there's any confusion because these are, I'm speaking a little fast and there's a lot of fine detail that has actually made all the difference in the findings that they have finally published. So let me know if there's any confusion at any point of time. In this experiment, they had bacteriophage cultures, which are obviously viruses, if you're familiar with the concept. These were bacteriophage cultures in which either the protein capsule was labeled with radioactive sulfur 32S, and alternatively, it was the DNA core that was labeled with radioactive phosphorus, and both of them were allowed to infect bacteria. So here you can see this is the 35S, I misspoke. 32P is the radioactive isotope of phosphorus. 35S is the radioactive isotope of sulfur. In this case, phosphorus will be able to only label the DNA molecule which is inside, as a result of which there is a non-radioactive coat. Yeah, there's a non-radioactive coat, whereas the DNA that is present inside the cell is radioactively labeled. In the other alternative, please understand they are both separate experiments. They were trying to figure out which part or which radioactively labeled moiety was actually injected into the recipient cell. So in the second experiment, where 35S or the radio-labeled isotope of sulfur was taken, only the coat was radioactively labeled, whereas non-radioactive DNA was retained within the bacteriophage. Now, both of these were allowed to infect bacteria, and then infect as in they were allowed to, they were mixed. Basically, bacteriophage was introduced in a culture which contained these bacterial cells. And if you look at this injection step, let me zoom in a little bit, you'll be able to see that for 32P, the red or radioactively labeled DNA is injected, whereas for 35S, the radioactivity is retained within the coat, whereas on the non-radioactive or the control DNA was injected into cells. Following, oh, sorry, just a second. Yeah. So as a result, when agitation was carried out in a blender, it was able to dislodge the phage particles from bacterial cells, obviously because it is not any sort of a covalent interaction. It's a weak interaction that occurs between these viruses and the bacterial cell, as a result of which the coat was separated and what was retained in the pellet, it contained our cells. So centrifugation allows us to concentrate these cells, separating them from the phage particles left in the supernatant. Now, when both of them were separately analyzed, it was seen that radioactive sulfur is predominantly found within the supernatant. However, radioactive phosphorus was found predominantly within the cellular fraction, which means that it had gotten introduced into the cells. And from there, a new generation of infective phage molecules can again be isolated. The conclusion, therefore, was that the active component of bacteriophages, which is able to transmit this infective characteristic, is indeed the DNA. And there's a clear correlation between DNA and genetic information. Furthermore, the injected cells, or the infected cells rather, are also able to generate new viral particles, which they are able to do because the information is retained within the DNA. From that DNA, more and more viral code proteins can be produced, which can package into new viruses, exit the cell, lyse the cell, and exit the cell, and can generate a whole new population of these infective viruses. So that is the basic idea, which allowed them to conclude about the nature or the biochemical nature or the biochemical properties of the inheritance molecule, which was the DNA. Taking this story forward in the 1950s, again, take a look at the timeline. So initial discovery by Frederick Miescher, very preliminary observations were in 1868. This were taken forward by the Streptococcus experiment by Avery, MacLeod, and McCarthy in 1944. Independently, Frederick Griffith also contributed to the same around this time. Then in 1952, which was pretty close to that time, this is actually the time where DNA was very actively being studied by multiple research groups across the world. And therefore, you'll find a lot of research which is contributed somewhere in the 50s and 60s around this time. Sorry, excuse me. Simultaneously, in the 50s, there came the large impact, or the very commonly known experiment, which is X-ray crystallography, that was contributed by the scientists Francis Crick, James Watson, Linus Pauling, Maurice Wilkins, and of course, Rosalind Franklin. So in the 1950s, Francis Crick and James Watson, they collaborated and they determined the structure of DNA. This is what literature will tell you, but of course there are many ifs and buts to this. But the key discovery, it is, or the Nobel Prize for this discovery is attributed to these scientists. So that is who we are crediting in this case. So they first revealed that indeed this is the structure of DNA. It forms this helical structure that allows DNA to maintain major grooves and minor grooves. And this was the conclusion or the analysis was the result of the discovery by Rosalind Franklin, which you can see in this image in photo 51, whereby she had used X-ray diffraction methods to understand how DNA is actually how the three-dimensional structure of DNA actually looks. And in 1962, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize in Medicine for this remarkable discovery. So, this is about the discovery part. Let us now get into details about how the molecule is actually formed. If you have some basic background about this, you would already be familiar with it, that the key molecule or the key uh, building block of DNA are nucleotides, or rather nucleotide triphosphates. So here you can see there's a sugar phosphate backbone to which a base, which is A, T, G, or C, is attached, that forms the nucleotide triphosphate molecule, commonly known as a DNTP, because this is for DNA. Of course, for RNA, that is different. There is a difference in the bases. There's a difference in the sugar phosphate backbone as well, altogether contributing to a different molecule. Second, you can see that Second, you can see that these individual moieties, they are forming a strand of DNA where they are joined by their sugar phosphate backbone. Bases project towards the inside, and templated polymerization of the new strand takes place during replication, and also during other processes where some bases might have to be inserted. When I say other processes, it is majorly transcription, but of course that's not between two DNA molecules. It is when a DNA RNA hybrid is formed, and the message within the genetic code is read, and with respect to that, an RNA is synthesized, and then from the RNA protein is synthesized. All of these processes take place in a similar manner. Then double-stranded DNA, which is present, consists of a sugar phosphate long chain backbone where hydrogen bonded base pairs are present on the inside, and then they eventually form this DNA double helix where you have major and minor grooves where because it does not form a very equal helix, it forms in a way that grooves are present to which transcription factors are able to bind, and they are able to read the information in that DNA, and then they are able to execute the downstream action. Uh, Additional discoveries which helped in understanding of the DNA were contributed by Shargaf as well. Shargaf's rules very commonly have been studied and are very crucial to the discovery of DNA. First of all, the idea that DNA in all organisms is the same is not true. The base composition very strongly can vary from one species to another. When we say composition, it is with respect to the idea that the prevalence of a particular nucleotide base is can actually vary from species to species. The same goes for amino acids as well. If you have studied about translation, the process of translation, then there is a preference of certain or the enrichment of certain codons within certain organisms. So codon optimization has to be performed in that case because a certain type of tRNA is more prevalent in that organism. Again, this is a different concept, but if you have any confusion, please let me know. I'm just discussing this, assuming that you have studied it at some point of time. So similar to that, for each organism, or for when I say organism, I mean however, when you see individuals from the same species, and when DNA is isolated from different tissues within the same species, they are seen to have similar base composition. Third, he said that the base composition of DNA in a species does not change with an organism's age, nutritional state, or changing environment. In all cellular DNA, regardless of the species, the number of adenosine residues or A residues is equal to the number of thymine residues, which is A equals T, and the number of guanosine residues equals cytidine, which is G equals C. Shargav basically comprehensively explained this finding as A plus G equals T plus C. So Shargav's rules were actually instrumental and had a huge contribution in establishing the 3D structure of DNA, and he actually showed the possibility that genetic information is retained and is maintained in a stable manner because of this nature. So, so the reason that this is so relevant and so considered so instrumental is because it explained to us that because there are hydrogen bonds that are forming between AT and GC, this is allowing the molecule to rest in an energetically stable state and allowing DNA to indeed function as a heritable molecule that does not change that does not change very easily based on the stresses or the environmental fluctuations, which is in contrast to what was already studied about RNA, which is a lot more labile, lot less stable, very easily degraded, and therefore proved that DNA can actually be the master molecule and can surpass the RNA world hypothesis. So this was the basic idea. So if you have to summarize the Chargaff's rules, then this is basically the rule that explains all of his findings. A plus G equals T plus C. How we understand it today is that there is DNA-based complementarity that allows DNA to maintain its structure. This was actually taken forward by Watson and Crick. You probably are familiar with this as well, the Watson-Crick model for DNA structure. So in 1953, again, see how close it is to the previous findings, whereas the initial gap was of about 60, 70 years in research about DNA. So in 1953, based on this available data, which included X-ray diffraction pattern and Chargaff's rules, Watson and Crick postulated a three-dimensional model of DNA structure, something like what you're seeing on the left here. First key property was that there are two helical chains that are wound around the same axis to form a right-handed helix. Now, what this means, I hope you already understand that there is a handedness to the helix. Simply put, if you curve your left hand and your right hand, you will see a difference in the way in which the axis moves. In case of the DNA that exists predominantly in Homo sapiens in our species, it is a right-handed helix because it curves in that direction, taking the thumb as the axis. In some very rare cases, you can read more about it from maybe Leninger's principles in biochemistry, where you'll understand there are A form, B form, and Z forms of the helix. In our case, we have a right-handed helix. The hydrophilic backbones of alternating deoxyribose and phosphate groups are on the outside of the helix, facing the aqueous medium, whereas the furanose ring of the deoxyribose, uh, whereby the furanose ring of the deoxyribose is in a C2 prime endo conformation. This, if you don't understand, don't worry about it too much. These are very key biochemical features of the DNA, but basically it talks about the conformation or the stereochemistry behind the organization of the backbone sugar phosphate backbone chain within the DNA molecule. The purine and pyrimidine bases on both strands are stacked towards the inside of the double helix with the hydrophobic part and nearly planar ring structures very close and perpendicular to the long axis. This is how you can see the arrangement is made. The pairing of the two strands creates a major groove and a minor groove on the surface of this duplex. This was the basic structure. Here you can see how far the bases are stacked. The width of the DNA is approximately 20 angstroms, whereas the entire one complete turn is around 36 angstroms, and this was actually called, I think it was called the step, which is the difference between two consecutive bases, that is around 3.4 angstroms. So it is not exactly 10, but around 10.5 bases that complete one full turn of this entire axis. And within this turn, you can see that this is the major groove. How will you understand the major groove? You can see that this cavity that is made by the turning of the helix is forming a major area for transcription factors to bind, and this is the minor groove for some other proteins to bind. So not going into details about it, but basically understand that this twist of the DNA creates a cavity or the scaffold on which certain proteins can bind. Now, how they'll bind, whether there's a sequence specificity, whether there's a conformational specificity, all of that is something we'll learn along the course when we discuss DNA protein interactions, which is quite a bit far from now. But for the basic understanding, I think this will be good enough. However, you can, of course, read as much as you want about it. There's a lot of information out there on the internet. But apart from that, for basic reference, for textbooks from which you can study from, most of the information that I've put in today's slides are taken either from Albert Leninger, or they have been Principles of Biochemistry by Albert Leninger, or from Molecular Biology of the Cell by Bruce Alberts. These two books are what I've referred to, and apart from that, some images have been taken from the internet, from review articles or from Britannica. So you can refer to those as well. I'll put the references for you later for your reference. Okay, moving forward, when we talk about the genome. So till now, we have just talked about the DNA, its individualistic features, what it is made of, its composition, and its discovery. If we just look at that, it is incomplete information. And what we need to understand in more detail is, if we look at a cell from a bird's eye view, what will we see inside? How does DNA look inside that cell? So to understand that, we will look at this entire architecture that you can see, which ultimately leads to the formation of a chromosome, which is what you can exactly see inside a cell. You can never see a helix because A, it is too small, B, we do not have microscopes that are of this higher resolution, and third, because DNA will not be stable in this unbound, loose, unstructured format. So before we move forward, let us understand the definition of a gene. This is something you can very commonly be asked in any interview, for whether it is for PhD, whether you are starting off your master's, or you're trying to apply in a lab for some project fellow, JRF, etc. This is a foundation that you should know and have a lot of clarity on. You'll find a lot of definitions of a gene. You'll find different ways in which it is reframed. But I think this was NIH's definition, whereby they call gene as an unit of hereditary information that has a fixed position or a locus on a chromosome. This is a compact definition. There are, of course, more additions you can make to this, but this is something that is almost always obeyed by a gene. Now, coming to the details about architecture, as we were previously mentioning, what we have studied till now is this initial extremely resolved organization of the DNA. So what you see here in colors are the ATGC base pair. This is the phosphodiester backbone. The approximate width of this has been shown to be around 2 nanometers, which then undergoes a lot of folding. If you look here, you will see that gradually we stop seeing this clean organization or this clean openness of the DNA, and instead a thick thick architecture taking place. Why does this happen? This happens because there are certain proteins present within cells called as histones. Histone proteins are DNA binding proteins that do not function as transcription factors. Their sole job, or not sole job, but their key job is to structure or to organize the chromosome in such a manner that A, it is stable within a cell, and B, it is compact, does not unnecessarily consume space and energy within the cell to sustain, and C, it helps in keeping DNA protected from digestion of nucleases and other enzymes, or degradation, mutation, etc. So it offers some structure and some stability to the genetic information within the cell. So each nucleosome, what is a nucleosome? Nucleosome is this unit which consists of DNA bound histone octamer. This octamer consists of 8 histone proteins, as the name octamer suggests, around which DNA is wrapped around 1.65 times. Please keep in mind that these decimals have, or these fractions have come up because of a certain measurement that has been carried out. So if you take a certain stretch of DNA and observe how many nucleosomes are present within the same, and how much of the DNA is wrapped around each nucleosome, it comes around a fraction. But obviously, that's not how exactly it happens. It happens because of the way because of the size of the histone octamer and the way in which DNA can actually wind around it. So DNA gets complexed with histones to form these nucleosomes. This nucleosome has the histone octamer core, which consists of 8 octamers. We'll learn all of this in a lot more detail when we discuss epigenetics. Don't worry. Epigenetics itself is a huge topic. We'll look at some key concepts, and there we'll learn a lot more detail about histones and the way in which they are actually able to structure these interactions. It doesn't look as It is not as simple as it looks here. There are a lot more interactions that are keeping it stable. Histones are not they're not just dead proteins within the cell. No, they are actually able to move about. They're able to control how much of the DNA should be opened, how much of it should be protected based on the situational requirement, whether or not any other process requires the histone to be moved. All of that regulation occurs within the cell. So we'll learn all of that. Do not worry. If we look at the dimension in this case, it's approximately 11 nanometers. So if you see here from 2 nanometers to 11 nanometers, we have wrapped up a lot of DNA within this place. H1 histone sort of acts like a stopper, which indicates that no more winding will take place around this nucleosome. And again, DNA can relax and start getting wound around the next nucleosome. So together, this is often called as a chromatosome. So nucleosome plus DNA together is called as a chromatosome, which consists of when I say histone, it also includes H1 histone, just for clarity. So this chromatosome consists of a nucleosome plus this H1 histone, which then again undergo another round of further folding, which you can see here. So how does this occur? There are again two ways in which this can occur as well. In a solenoidal manner, this representation has been shown. We'll look again at all of these topics in a lot more detail later. But for now, see that the chromatosome arranges itself in a manner where all of these repeats are stacked one on top of the other to produce a 30 nanometer fiber. So as you go from 2 nanometers to 30 nanometers, you can see that so much of compaction has taken place where from a open thread of DNA, we have now wound it around these nucleosome moieties, tried to accommodate it within a smaller space so that it can be sort of squished and arranged within a limited region. Following this, there are loops that are again formed. These loops then form around 300 nanometer wide stretches. These loops further undergo folding where they undergo more and more compaction where they form a 250 nanometer wide fiber. Then their tight coiling finally ends up producing the chromatid of a chromosome. So what you see when you look under the microscope for any sort of experiment like say for example onion root tip cells undergoing cell division or buccal cells or bar bodies for example, imagine the amount of biology that has been studied, that the cell has undergone to compact all of that information for you to be able to see it as a chromosome. So it's incredible, and it's something that is actively occurring in every cell that is undergoing division at any point of time, because this information needs to be compacted every time a cell undergoes the process of cell division. So I hope it was clear. I think I might have spoken too fast, but if it's something that's unfamiliar or difficult for you, please let me know. I'll be happy to try and explain it better. Okay, so moving forward, if any doubts are there, please interrupt me. I'll try to cover the topics so that we can, wrap up whatever needs to be done for today. So, this was how the genome is organized within any cell, for that matter, any cell where DNA is arranged into chromatids on a chromosome. Coming to the human genome at large, if you take the snapshot, this shows you that if this is the entire genome, from there, a lot of information is something that we don't even take into consideration when we are regularly studying things. What do I mean by that? What I mean is that we mostly look at the genome as something which encodes an RNA, which encodes a protein, that is the central dogma of biology, and therefore our major focus is very unfortunately restricted only to protein coding genes. However, if you look at it from a bird's eye perspective, you'll see that that takes up a very small percentage of the genome, which is around 1.5%. And that gives us around 20,000. Now it is considered to be somewhere around 28,000, close to 30, but around 20,000 protein coding sequences, which are interspersed by non-coding introns that make up a much larger fraction, around 26%, Transposable elements, on the other hand, make up the largest fraction, around 40 to 50%. If you're not familiar with it, transposable elements are often called as transposons, which are jumping genes, which are sequences or fragments within the genome that are able to move from one location, one chromosomal location to another because of their nature, because of some repeats that are present. They are considered to be jumping genes. We'll discuss this also in more detail for non-protein coding genes later on. I think somewhere in the third or fourth week, it is probably there, where we'll learn about the discovery made by Barbara McClintock and how relevant transposons are in today's time and what have we done as human beings with that information that was discovered so long ago. So anyway, transposable elements, they take up this large fraction and they include lines and signs. Lines are long interspersed nuclear elements, which I think is here, yeah, around 20% of the genome. And short interspersed nuclear elements are signs, take up around 13%. So most transposable elements, they are genomic remnants, which are currently not functional. And remaining, you can see some are segmental duplication, some are miscellaneous heterochromatin, etc. But apart from that, DNA transposons and LTR transposons also take up the remaining region. So most of the genome is actually not even coding for the proteins that we have within the cell, which makes it look almost redundant. But of course, if you know biology, you know nothing is ever completely redundant. Everything has almost always is going to have some function within the cell. What might happen is that we are not familiar with that function and hence our limited knowledge regarding it. So currently what we are going to focus on are these protein coding genes. And what we are going to keep in mind is that most of these remaining regions of the genome are contributing very largely to the regulation of the genome and regulation of the protein coding regions that are present within the genome, both cases. So with this idea, we move forward. Um, what you see here is the complete set of human chromosomes. These have been stained, artificially colored, but they have been stained to show how the chromosomes actually look within the cell. You can observe that there is visibly a lot of morphological difference between these chromosomes, whereby some of them are longer, some of them are shorter, some have a sort of kinked architecture, and some are completely straight. Some are very small in size, like chromosomes 21 and 22, whereas some are quite large, like chromosome 1. Uh, all of this information is quite variable within the cell, but they have a large role to play. And the size of these chromosomes also makes a lot of difference in the propensity of different chromosomes to have different sort of events. All of that is a far advanced concept. You don't have to worry about it, but I just wanted you to, sort of gain perspective that when we started from here, from DNA, to reaching this entire, going through all of these steps to finally reach the structure of a chromosome, which is what you see here, there's a lot that has happened. And finally, this scale bar is going to help you understand that a little better, that all of this information got compacted into such a small region of only around 10 microns, and that is what you're able to see with your naked eye. Not with your naked eye, but with the help of a simple microscope. So there's a lot that happens that we don't even think about. Moving forward, we were talking about the central dogma of molecular biology. So, like I mentioned just on the previous slide, that the default hypothesis about the central dogma is that it's considered to be a theory that states that genetic information flows only in one direction, that is from DNA to mRNA to protein, or from RNA directly to protein. If there is an existing RNA, then from there it goes directly to protein, or it goes through transcription from DNA to reach this state. However, we do not consider this absolute. That is why this is still called as the dogma and not a confirmed fact. That is because there are many ifs and buts about it. First of all, an interruption that occurs is in this very first step, whereby reverse transcriptase enzymes are able to do the exact reverse of the same, where they can convert previously existing mRNA into DNA because of the way in which that enzyme functions. And this is very actively used in a very common experiment within any molecular biology lab that you might know, where we carry out cDNA synthesis from mRNA that we isolate from cells, and then use that cDNA for quantification by qPCR. So, what exists in nature has been sort of utilized and modified and utilized by us for our own, own utility. Additionally, some other changes also that have been made over time include the fact that DNA undergoes its own replication. That is also, in most cases, included as a part of the dogma now. And also the fact that sometimes proteins that are able to function as pathogens, like prions, they are also considered to be an exception to this theory, whereby they do not exist as they do not have to undergo all of these steps, and they're able to independently carry out infection of the organism in which they are present. So there are some ifs and buts to this, but the basic central dogma of biology that almost every protein coding gene follows involves the conversion of DNA to RNA via the process of transcription, and then from the mRNA to the functional protein, which is by the process of translation, and then the polypeptide chain that has been synthesized will undergo proper folding, which in a large number of cases is also co-translational to produce the tertiary structure of the final protein that is able to execute its own activity. So this is the basic flow. And apart from that, what you see here is the basic structure of a gene. That is because if you move from the DNA to mRNA to protein, you should also understand that how and what is the way in which its architecture is, or not the architecture, the organization of the gene actually makes the difference. That is because the region that actually undergoes the process of transcription and translation is pretty small. The rest of it is regulation, and that regulation is quite extensive. Let's take it one at a time. First of all, I just mentioned the region that undergoes downstream processing, which is the open reading free, or the ORF, which is present for each and every gene that encodes for a functional protein. So, what you see here are exons that are regions that will be retained, and they are interspersed or interrupted by introns, which are commonly also known as interrupting sequences. And it is flanked by a poly A signal sequence towards the end of the last exon, which indicates that this is where transcription should undergo termination. Following that, in a large number of cases, terminator sequences are also present, which ensure that transcription does end right after this point. And additionally, we also have upstream and downstream regulatory sequences. In this case, upstream regulatory sorry, upstream regulatory sequences include some proximal and distal control elements. What does it mean? Proximal means something, means those sequences which are very close to the ORF, which are very close to the start of the gene, and they are almost adjacent to the promoter. So the promoter containing the Tata box is almost in contact with the proximal control elements. However, distal control elements are those which are present at a distance, which are far off, but are able to execute their activity by chromatin folding or chromatin looping, which brings very far regions close to the promoter and help in controlling or manipulating how DNA expression or how gene expression should be carried out. So these distal elements often or are more commonly found to include enhancers or silencers. Enhancer is something which increases the expression of a gene, and silencer obviously silences the same. Also, keep in mind that these distal control elements can either be present as upstream regulatory sequences or downstream as well, which means they can be present 5' or 3' to our gene of interest anywhere within the genome. And it can be brought close to the controlling regions by chromatin looping. So, this is kind of the structure of a chromosome. Now, this is again okay, all these images that you're seeing, this they are all part of Bruce Albert's molecular biology of a cell, so you can refer to the concrete text from these books also, so your own reference for brushing up these topics. So Yeah, this image on the left shows you the basic, gives you a perspective of how a gene is actually organized within the chromosome and within the organism by large. So here you see human chromosome 22 in its mitotic conformation composed of two double-stranded DNA molecules, each around 48 million nucleotide pairs long. So that is huge. 48 million, imagine the number that is present in each each DNA molecule in each chromatid. So yeah, this is what you see here. This is the heterochromatin. And if you expand this region, then you will see that around 10% of the chromosome arm consists of 40 genes. If you zoom in further, for example, within this region, you will see that this is 1% of the chromosome arm that contains only 4 genes. Again, take a look here to see how tiny the chromosome 22 is compared to much larger chromosomes. So the enormity of the genome is actually apparent only in these circumstances because 22 is rather small. If you look at chromosome 1, chromosome 2, they are immensely large, and therefore the amount of information retained starts making a lot of sense. So anyway, within 1% of this chromosome arm, if you zoom further, which contains 4 genes, if you zoom further, you will see that it contains some regulatory sequence, which is, of course, helping in controlling the gene expression. You have several exons shown here in red, and you have introns shown in gray. And again, within this population as well, you'll see that introns predominate over exons. However, that is necessary to control gene expression, to control how transcriptional regulation will be carried out, how certain regions will be retained, whether there can be alternative splicing, whether there can be modifications, whether there can be control, etc., etc. So this ideal situation, it would involve the joining of all of the exons or the retention of all of the exons and the removal of all of the interrupting sequences or the introns, followed by generation of the mRNA by the process known as transcription, which is